Finally, we got the lab going, so we're excited to start doing stuff. Originally, I'm from Nigeria, born and raised. I moved to England, and then I moved to Ohio. From Ohio, I moved to Wisconsin, and I studied applied microbiology and brewing years ago. So I remember the first time I brewed beer back home, and my brothers watched the whole thing. I finished, and it was evening. And when I finished and I was gonna drink the beer, they said, "Well," I told them, "Do you guys want to join me?" They were like, "Well, don't finish it. Drink a little bit. Keep the rest in the fridge. When we wake up in the morning, if you're still alive, then we'll join you and finish the beer for you because we're not sure we're gonna make it on this beer you just made yourself." So anyway, I drank a little bit, a whole lot, in fact. Went to bed. By morning, the beer was gone. As soon as I woke up, they said, "Oh, he's alive." My brothers went and just shocked everything off. So I've been brewing ever since then. I haven't done that lately. I'll be talking to you a lot more today about new yeast in brewing and the prospects for new flavors and the related challenges with this process. Now, for every brewer, one thing we're very passionate about is consistency. You want a situation whereby whatever you brew every time has the same flavor, aroma, body, and character. You don't want that to change one single bit. This is even more important for commercial brewers because you don't want a situation whereby someone gets Heineken and they drink it tomorrow, they feel like they're drinking Milwaukee Brew or some other brew, like, what is this? Now, it doesn't mean there's something wrong with it, but the fact that the flavor and aroma are not the same, they tend to think there's something wrong with it. So that's why consistency is a very, very big thing in brewing. Whether you're making IPA, whether you're making wheat beer, whether you're making brown ale, whatever, you want to retain the same characteristics all the time. Now, that's why branding is a big thing in beer. And I think branding is not all about the keg or the bottle or the can. It's what's inside of it. The beer itself is what the brand is all about. If it changes in taste and flavor, there's something wrong with it right away. And for that reason, we've used two major strains or species of yeast to brew over the years. Saccharomyces cerevisiae and Pastorianus. As a matter of fact, both of them account for over 90% of all the beer brewed worldwide, because we have that desire for consistency. We want to make sure that each time we brew, we get the same characteristics. You don't want to change the organism you used to do that. Also, it's a very traditional industry. All the condiments have to be the same all the time, the same amounts, especially the yeast. You don't want to switch that at all. We can actually engineer organisms to produce particular flavors, try to change the amount of alcohol they produce and things like that. That's what most of my work is. Not necessarily for beer, but for other things. We switch the genes in yeast and try to make it to think in a different way and do different things. But for brewing purposes, that has a huge market perception. People will start wondering, that's GMOs you're giving to us. How are we sure that's not going to affect us in the future? So it's not something we do a lot in the brewing industry. So for that reason, we're very much committed to brand image and customer loyalty, making sure that what we pump out every time is the same all the time. However, from a scientific background, we are gaining a lot of understanding of how yeast behaves. We're understanding the genes that make up yeast. We understand the genes that are responsible for alcohol production, flavor production, and so on and so forth. As a result of this pool of information we're gathering, there's a possibility for us to be able to make some switches, possibly, not just from one organism to the other, but also within the same yeast species. For instance, you can look at this yeast here, it's the same organism, Saccharomyces cerevisiae, they all look alike. But genetically speaking, just like two siblings, born of the same parents, they could be even identical twins. If you look at the genetic makeup, you find out that one of them will be very significantly different from the other one. So if I look at one particular cell out of this pool of Saccharomyces cerevisiae, you find out that one of them might have a slight mutation somewhere that could enhance the production of a particular flavor that gives me a particular brand of beer that's going to be liked by a lot of people because of the flavor I'm producing. However, without genomics, you don't get to see that. Today, we're able to look at one species and dissect it. And even within the same species, we can see differences that could be used for brewing applications. Also, we can look at non-Saccharomyces cerevisiae yeast, so to speak. Rather than focusing on just one yeast, genetics allows us, or genomics allows us to look at different organisms and to be able to understand the genetic constitution, which tells us some of the flavors they produce. Also, you find that there's a process called hybridization, which has been around for a very, very, very long time. but hybridization often is talked about as a natural process, which I'll talk about a little bit more. However, if I understand the genetic makeup of one organism, I can take that organism and hybridize it with another. Now, first of all, this happens naturally. That's how Saccharomyces pasteurianus was actually formed. It's a natural fusion between Cerevisiae and Saccharomyces eubayanus. Two of them fused together to form a different strain called pasteurianus, often sometimes called Casbougensis. Now, you find out that most of the characteristics of this particular species now are born out of eubayanus. However, if I understand how one strain behaves or one species behave, I understand how the other behaves. Now, one of them makes high alcohol, for instance, one of them makes flavors. I could use hybridization to fuse two of them together to create a new strain, not necessarily genetic modification, because we've been using this for a long time to brew beer, and it's a fusion between two different yeasts formed naturally, and we've domesticated them. You wonder, can we do the same thing where we can hybridize two different strains that have characteristics that we want together? One makes more flavors, the other one probably makes more alcohol. If we fuse them together, can we get a strain that can enhance beer production in terms of alcohol content and flavor as well? Also, based on our knowledge of genomics, we can adapt organisms. What we've done with Saccharomyces cerevisiae and pasteurianus over the years is just to domesticate them, subject them to conditions where they can use maltodextrin work much better, and they can produce higher alcohol content. Now, can we take other yeasts as well that have flavor characteristics that we desire, but don't necessarily make enough alcohol, domesticate them to be able to do what we want? Now, genomics allows us to do that, because with genomics, we can see, yes, this yeast has all the characteristics I want, except this. Can I naturally adapt it to be able to behave the way I think? Again, you're avoiding genetic modification, but you are adapting the organism to conditions that will force it, just like all of us, to learn to do what we want it to do. But you don't want to start with a bad strain or species in the first place. You need to understand what that particular species or strain does. And that's where genomics comes in. Now, this has been going on in the wine industry for many years. They are not as traditional as we are in beer brewing, so to speak, because they've modified a lot of yeasts over the years. They use them to be able to create different flavors in wine. And you wonder, is that something that could come to brewing in the future? Someday. That's a different debate at some point. But there's no doubt that people are demanding different flavors in terms of what they want in their beer. One example is the alcohol-free beer or low-alcohol beer. $9.6 billion industry. It's going to be worth $35.6 billion in 2030. And what is this all about? We're just making beer with low alcohol or no alcohol at all, but we want to retain all the flavors. How do we get those flavors? I'll talk a little bit more about this in a short while. And naturally, what we do with alcohol-free beer is we produce them by two different methods. One is physical removal. You produce the alcohol, you pull the alcohol out, retain the flavors. The other one is arrested fermentation, whereby you subject your yeast to conditions that will not favor alcohol production, such that they produce more of the flavors and not enough alcohol. But in reality, neither of them works very efficiently, because when you make alcohol, you pull it out, most of the flavor compounds are volatile. You pull them out with the alcohol as well. So you have less flavor, less alcohol. But so the taste is not the best. Okay, you use arrested fermentation. Now, yeast behaves like any other living thing. If it's active making alcohol, it's making flavor enhancing compounds as well. If it's not making enough alcohol, it's not making enough flavor enhancing compounds for you. So in that case, it's the two methods we have today are not ideal. And that leads us to ask the question, could we have better strains that could do this in the future, given the fact that this aspect of industry is going to grow a lot in the future? And generally, people are looking for more flavors in beer. I think all of us that are involved in craft brewing understand this. That's why the industry has grown. I think people have grown tired of particular brands and products over time that they demanded something different, and it's all about flavor. And that's why craft brewing, I think, is going to even grow going into the future. And the other one is hard seltzer. It's like a blank canvas. You brew whatever you want, open with sugar. You can create any flavor you want within that. Again, that industry has grown a lot over the years. And that leads us also to ask the question, can we look at other yeasts beyond the traditional ones in brewing to be able to create some of the flavors that can attract more people to our industry? So that begs the question, how can we meet this demand? What can we do about it? One way to do that is look at known or less traditional yeasts in brewing. and assess the capability to do some of the things we're interested in. One example is one I mentioned a while ago, Saccharomyces eubayanus is a fairly new yeast. Of course, it's been around, but we discovered it in 2011. It is the parent, so to speak, of Pasteurianus, which is used in lager brewing. It grows at low temperatures. In fact, it accounts for 99.5% of the genetic composition of Carbogensis or Pasteurianus. It's an unintentional fusion between Saccharomyces cerevisiae and eubayanus, as I mentioned a while ago. This yeast does not utilize maltotriose. I'll talk about it a little bit more as we go forward, because often we worry, if I introduce this into my brewing process, there's a possibility it's going to overrun the culture. That's not true, because if it's incapable of utilizing all the sugars in the system, it cannot be the dominant yeast strain or species within your system. But we'll talk about that a little bit more as we go forward. It doesn't grow at high temperatures, so you have means of controlling it as well. It doesn't produce as much ethanol as the more traditional yeast in brewing, so you have to rely on it mainly as an agent or catalyst for producing flavors. It does produce high fusel oils or fusel alcohols, which are very important in brewing. Also, it produces 4-vinyl guaiacol, which is important in craft brewing. It's responsible for that clove-like smoky flavor you get in beer. So it does have the capacity to make more of the flavors we desire in brewing, but not necessarily high alcohol content. So in summary for this yeast, if you look at it, it grows well at low temperatures, it's good for lager brewing. It uses maltose well, but not maltotriose. Now, you have to wonder, can we innovatively use this to brew something, or in combination with another yeast, or by itself? Or can we even hybridize it with an entirely different yeast species or strain to be able to create another yeast species that can do exactly what we want? As a matter of fact, some years ago, it was in 2011, Heineken actually put this to the test. They brewed a limited edition of Heineken in Argentina called Wild Lager, using Saccharomyces eubayanus, plain blind, nothing else, only this particular species. It was a limited edition. I'm surprised I haven't done that since then, but it was well received at the time. So this was done in Patagonia, Argentina. They were trying to see if the wild species can do something that the domesticated ones can do. Of course, they were able to get decent alcohol content, but again, this beer was full of flavors because of the capacity of this particular yeast. Now, if you're brewing, you have two methods you could actually go with, which the other one is less used most of the time. You have the conventional method. You control everything. You bring in your yeast and sugar and everything, brew the way you want. Or you have a non-conventional method, in which case you rely on the naturally occurring yeast to be able to do the brewing for you. Now, either way, you're going to get something either good or bad. Now, with non-conventional methods, you're likely going to get something bad because of off flavors if you're not controlling the conditions. But once in a while, you can hit on something based on the yeast profile there that will give you a final flavor profile that beats what you have via the natural or conventional process. So most of the time, though, people don't want to do that. The reason they don't want to do that is control. You want to control your brewing process every single step of the way. If I don't control it, I could get something bad. But most importantly, you want to collect your yeast and use that to repitch your next batch. Now, if I use two different strains where I think one is going to produce more of flavors for me, one alcohol, good. But what about the future? One of them might overrun the culture, so much so that I'm not getting exactly what I want because I'm not controlling it. That's one thing most people worry about, and that's a very legit worry, so to speak. But this is not the case because nature has given us, in some cases, depending on the yeast you're looking at. Now, a good number of the wild yeasts are not domesticated. They are not exposed to wort. They cannot ferment maltotriose, which is the second most abundant sugar in your wort. If they can't do that, they are less likely to become the dominant strain within the system. If they can't be the dominant strain, they cannot overrun your culture. So there's a natural means to control that if you're willing to try something like that. So think of it this way. You have your wort, you have your maltose, major sugar there, you have maltotriose. You have your domesticated species in this case, which can use both maltose and maltotriose. And you have the wild species that can only use maltose. In this case, it's almost impossible for the wild yeast to overrun the culture and become the dominant strain because it's incapable of using all the sugars there. The one that uses more sugars will become the more dominant strain. So there's a natural selection in place to ensure that the more wild or wilder yeasts do not become the dominant strain if you're combining yeast species or strains to be able to create desired flavors in brewing. There are other examples as well, like Chlorveriomyces maxiana, sorry. This organism doesn't produce a lot of alcohol, but it does produce important flavors: phenyl ethanol, 2-phenyl acid, 2-phenyl ethyl acetate, which is desirable in brewing. Also, you have Hanset Neospora species and Brettanomyces species. Often we call them contaminants in brewing because they produce off flavors, but a low amount to produce more desirable flavors in brewing. So you can use this, but the problem is control. Can you control them? Natural selection allows you to do that based on the fact that most of them don't like multi-trials. Also, most of them do not grow well at high alcohol content. If I'm blending any of the species with my traditional yeast, at some point they're going to die first. They die first when the alcohol composition rises. In that case, I'm selecting them out, ensuring that the dominant strain every single time is going to be my traditional yeast. Also, you have Pterolepis delbrueckii, which produces fruity amyl alcohol flavors. As a matter of fact, this particular species is resistant to alcohol. Doesn't produce a lot of alcohol, but is resistant to it. So it can become persistent in the system without overrunning the culture. It doesn't use a lot of multi-trials, doesn't use it at all, doesn't produce a lot of alcohol, but can tolerate high alcohol content, which means it can be active much longer to enhance flavor production in your brewing process. In fact, there are commercial preparations of delbrueckii available today, which I know some people that have used there. So most of these sugars, most of these yeasts, do not utilize the second most predominant sugar in brewing, which means they are there specifically for making aroma, and most importantly, they are less likely to overrun your culture. If you're making low or free alcohol beer, these yeasts are more likely to become important in the sense that they don't produce high alcohol content. If I don't desire high alcohol content, that is exactly what they do. But I desire the flavor of beer, and a good number of them can produce those flavors in brewing. So there's an increased demand for low alcohol brewing beers. As a result of that, I think this trend is a real opportunity for people to look at some of these yeasts going forward. And I do know a company I talked to recently are looking at the possibility of switching the process. They are researching this, I should say, so that they can go away from pulling the alcohol by vacuum or using arrested fermentation, but still be able to produce low alcohol content and retain the flavor of beer. And also, some people are looking for that sort of something weird or wacky in brewing, something completely different. They want a deviant, so to speak, kind of flavor in brewing. Most of us have more weird desire for flavor than others. A typical example are the acidic beers. You have the Lambic beer from Belgium. You have the Coolship ales in North America. You have the Berliner Weiss. These beers are predominantly acidic, predominantly because of lactic acid produced in them. They're not as popular, but they have grown in popularity over time. Now, most of the time you find out that that acidic sharp flavor is produced using lactic acid. But if you're a brewer, you know that you don't want lactic acid around your brewing because they grow faster than yeast. They can take over the culture. They can contaminate everything. So most of the time, those who brew this don't brew anything else to stick to their acidic brews, as a matter of fact. However, there are yeasts that can do the same thing without having the capacity to overrun your culture because they don't grow as faster than your regular traditional yeast. An example of the Brettanomyces species, you have Decara species as well. So if you're using these Brettanomyces and Decara species, you find out that you're less likely going to run the risk of having something contaminate your culture long-term because the yeast you're using grows either at a lower rate or at the same rate as your traditional yeast. And there's another yeast that's become very popular over the years called Lachancy thermotolerance. Now, it produces lactic acid. It doesn't contaminate your culture. It doesn't overrun your culture, but it produces lactic acid, which adds that acidic freshness to lambic beers. It doesn't utilize mortar trials, which is a good advantage, which means it doesn't have the capacity to overrun the culture. But it produces good flavors, but not off flavors. And I talked about Brettanomyces and Decara a while ago, which are good for making flavors, but if you have too much of them in the system, they tend to produce off flavors at high cell density. So for this reason, Lachancy is a much better candidate for making lambic beers because they can produce acids without overrunning the culture, and they don't produce off flavors as well. So apart from these naturally occurring yeasts that you can use as substitutes in brewing, there's also the possibility of using hybridization. Now, I did talk about that. It's a natural process where two different yeast species or strains can come together, fuse, and form a new, different, larger yeast that may have different characteristics that you desire, or combination of the characteristics from the parent strains or species. Now, while this occurs naturally, we can induce this in the lab as well, using spores most of the time from one species and another, blending them together under the right conditions to make a larger yeast with desired characteristics. As a matter of fact, a lot of people have done this. There's a study by Garcia, Sanchez, and Cole in 2012. They fused Saccharomyces cerevisiae and Pasteurianus, and they produced a number of characteristics like increased tolerance to higher temperature. They also increased tolerance to higher alcohol concentrations in the yeast strains. And again, you're not genetically modifying each of the strains or species, you're fusing them together. And bearing in mind that Calvagensis or Pasteurianus, which we already use in brewing, that's exactly how that occurred. So you're doing something that nature does, except that you're being the one bringing two different species together. Now, there's a study as well by Stencils and his colleagues. They produced about 46 different hybrids from 3 different genetically diverse Saccharomyces cerevisiae strains. So they got 3 strains of Saccharomyces cerevisiae, fused them together in so many different levels and ways, produced 4 to 6 different hybrids. And they came up that, so they saw a lot of characteristics at the end, but some of the most important ones were that some of them produced 3-methyl-butyl acetate, which adds that banana peel aroma to your beer. And there's another process as well for enhancing the characteristics of your yeast. Now, you can either use hybridization, which often uses spores, you fuse spores together, or you can do what we call protoplast fusion. With protoplast fusion, you're not working with spores, but you're working with your yeast cells. This is an age-long process that's coming back of late. What you do here is you can get one yeast, get another yeast. You use an enzyme we call lysozyme in the lab to remove the cell wall. If you remove the cell wall, they get sticky. If you add them to PEG, polyethylene glycol, the PEG makes them even stickier, that you get them together, 2 of them can fuse and form a bigger yeast species, or strain in this case. But the point is, to be able to do this, you want to know what you're fusing together. With genomics, you can have pinpoint accuracy, knowing I know exactly what this strain does, I know exactly what this other one does. I can pull the 2 of them together to get a particular characteristic that would or could enhance my brewing process. You fuse them together and you get a much larger yeast most of the time that could have the characteristics you want. This process has as well been used by Choi and co-workers in 2002, long while back, and they produced a yeast that's capable of using dextrin. Of course, most of us know that dextrin is a much higher chain of sugars. Now, if you're fermenting dextrin, you can cut down your costs, as a matter of fact. Rather than hydrolyzing your sugars down, you can use dextrin, in which case you reduce the process cost and the process longevity itself to be able to produce your beer. And they were able to demonstrate that this works very effectively with this, the resulting yeast strain they produce after fusing the 2 strains together. Now, it doesn't always work. Sometimes you have hybrid sterility, especially if you're working with spores. You want your spores to be potent over time so you can pick them and fuse with new ones. For those who do this, sometimes you do experience sterility, not all the time. If you do, most of the time, what people want to do is to maintain their cultures over time. You don't have a need for making spores. As a matter of fact, the traditional yeast we use in brewing don't make spores. They've lost that capability over time. And then you can also have this integration with fused cells. This often happens with protoplast fusion, where you fuse 2 yeasts together. Now you have to select and test them over time, over several generations, to ensure that they stay compatible together. Now, some of them will disintegrate, whereby they literally reject each other. One way to think of it is like someone getting an organ donation, and then you put it in the person's skin, and the body rejects it. It does happen sometimes that you fuse 2 different strains together and you find that they reject each other. So, but to be able to avoid this, you want to first of all fuse, test over a long period of time, several generations, up to 50 to 60 generations, to ensure that they're able to stay compatible and don't break down. Otherwise, you can start something with a particular strain, and over time you don't have your strain anymore because they're disintegrated. So these are 2 major challenges with the process of either hybridization or protoplast fusion. But for sure, you can still select strains out of this that can stay active for a very, very long period of time. So in general, people are demanding a lot more diversity in brewing. People understanding how brewing works. They're also understanding that yeast plays an important role in the flavor we get in beer. They're more interested in it. It used to be just give me beer. There are more and more people involved in brewing today because they're interested in craft brewing. And I think craft brewing has brought beer much closer to people. As a result of that, people are demanding new styles as well. They're demanding new flavors. And I think to be able to address that, going into the future, there's a possibility of looking into genomics as a tool to do that, whereby brewing industry can work with scientists via research to be able to understand how we can select the right strains, the right species, even within the same species, to pick out the right strains that might have particular characteristics that we want. But most importantly, there's also the possibility of fusing strains and species together, possibly to get much better characteristics over time. So I think these technologies over time will play an important role in how brewing shapes up in terms of flavor particularly. And also I think there's a possibility of domesticating new strains or species over time to be able to meet that demand for new styles of brewing, for new flavors and what have you. So I'd like to thank you all for having me, and I'll take your questions.
Okay, well, Victor, perhaps I can start with the questions here. I'd like to talk about risks because brewers tend to, as you so rightly said, we tend to be a little bit we're not too much of risk takers. And you were talking earlier about 4-vinyl guaiacol. Whether we speak about 4-vinyl guaiacol or whether we speak about 4 sort of ethyl guaiacol, those are two, at least in my experience, very undesirable phenolic flavors that we don't want in our beers. So I agree that certainly some of the things which you presented will be used by the brewing industry eventually, but I think that since we're conservative, we probably don't want to go too fast, and then there's a risk of certain sort of off flavors that could occur, which will make people nervous about using some of these new strains. What do you think about that?
I do not think that change is going to happen overnight at all. Anybody who brews is very traditional, including myself. I don't think we'll wake up tomorrow and have an entirely new strain for brewing, but I do think down the road, if you look at 10 years ago, 20 years ago, craft brewing was not as popular as it is today. And you talk about hard seltzer, you talk about low or free alcohol beer, not as popular as they are today. So there's been a sort of change in the industry. And at some point, I think someone's going to figure out how to be able to create new flavors that are appealing to people using new strains or species that we're not even looking at now. Until someone proves it and shows that it works long-term, it's still out there for now. I think the jury is still out on that, but I do believe that at some point it's going to happen. It may not happen as fast as you rightly said, but there's a possibility that most of the newer, less tested strains could have the potential to enhance brewing flavor-wise over time.
Okay, thank you, Victor. Because I think that when you started off your presentation about talking about non-alcoholic beers or near beers, whatever, I think there's very good reason to be able to be using strains which do not produce alcohol but do produce, as you said, these different flavor compounds that can really complement the beers that we currently have. So I think that can be very exciting. And in terms of the popularity of something like Heineken 0.0, it's big business now. So it's going to be amazing, I think, when you can use these strains that don't produce ethyl alcohol but do produce all these different flavors that perhaps the customer would like to have in a beer.
I agree with you. It's likely going to happen, but again, it's going to take quite a lot of testing to ensure that you can consistently produce those flavors without having off flavors. But I do know a number of people are looking at that now.
Thank you.
You're welcome.
So I'm kind of curious, you talked a little bit about the some of these strains and not being able to produce maltodextrin and therefore, them not being able to become dominant strains. I'm curious if you've either you or you know of people who've done pilot trials as far as seeing whether or not the opposite happens. So you might introduce this yeast to try to get some of these flavors and they don't become the dominant strain, but then they become outcompeted and don't really contribute. Have you looked at all into how far that tends to go before you could kind of lose that impact with different strains?
The few studies I've read about that, you find out that they do hang around the system. Whenever you have two or three organisms together, the dynamics are more complex than you would with one organism. Often one of them will try to outcompete the other. But what happens is that if you have maltose, you have maltotrials, and you have two strains, and one of them can use both. Often they don't use both at the same time because it's not economically or biochemically economical for the cells. Like you have a plate of rice, a plate of beans. Why eat both at the same time? You want to finish one before you eat the other one. So for that reason, you find out that often one of them tends to focus on one until they can get to the other one. For yeast, most of the time they go for maltose before maltotrials. For that reason, you find out that if you inoculate in a particular ratio over time, you're likely to keep the dominance of your desired strain constant over the other one. Now, whether your dominant strain or preferred dominant strain overruns the other one, for sure that could happen, but it's down to how much of the wild strain you're pitching into the system. If you're pitching too little of that, it doesn't stand a chance. Now, if you're pitching, let's say something like 80%, 20%, it does have a chance of actually producing flavors within that period of time before maltose runs out and your dominant strain goes onto the maltotrials. So it could happen, but it's down to the inoculation ratio or proportion you're putting into your pitch.
Sure. And I guess what I'm thinking more about is just subsequent generations. So say you pitch 80-20, and then the next fermentation is it something like 85-15, and then the next one is at 90-10, could it go that direction as well? I mean, I guess it definitely could, but I'd be curious what the trends are.
It could. It's one of those things that most people haven't really tried that on a commercial scale because you want to constantly test to see where you stand. And the investment associated with that is something that puts people off. Why should I have to test all the time? I want to put one thing, I want to get my result out all the time. But it's down to, can someone do it in a way that you get something that tastes superior? Then the incentives becomes that people want it so badly that you want to keep doing that. Probably there's a price tag for that. So in the absence of that, it's quite challenging to be able to try that. But there's no doubt that it could be done. But there's still a lot of research to be done to be able to understand how far you can push that, your generations over time, to retain the less dominant strain still within the system. So yes, it's a very good question. I think it could get run out over time. In which case you can keep repitching small amounts of that.
Well, that dovetails into my question. So on a practical level, if you're doing like a co-pitch in a tank, is there any way, I mean, would you use like cell counts to kind of figure out like, this is the percentage I have in this tank from strain A versus strain B? And would that just be like, is there an easy way that you could quantify that?
There is an easy way, but most people who brew don't have the tool to do it. It's PCR.
Okay.
Yeah, because if you're measuring the cells, the cell meter or spectrophotometer doesn't tell them apart. It just sees cells there. It cannot differentiate. Sometimes when you plate some of them out, they look alike, and it becomes difficult. Even if they don't look alike, it's quite labor intensive to plate out and count every single time. But you could do PCR where you test what we do quantitative PCR. It tells you the amount of DNA for strain A, the amount of DNA for strain B. Then you can always tell over time how much of them they are there. I've tried experiments like that in the lab a whole lot of times, and you can easily detect which is what at any point in time. Based on their genetic signature, it tells you which ones are growing there and to what extent they're growing, and that can give you an idea which one is dominant and which one is not. For the brewers, it's not something they find practical to have.
I guess my second question was, is there, knowing that the like, let's say the primary microbe is Saccharomyces cerevisiae, and then like the secondary microbe is some offshoot of Eubanus or something like that. Developing that secondary or alternative yeast or microbes for brewing, is there any way that people are building in some kind of control aspect? I just, as far as like the secondary microbe goes, I know that a lot of those are potential infections where, you know, a brewery like Octopi would be able to use this tank for a customer that they have that wants to use this microbe. Are the guarantees there that they can kill it so that the next thing that they put into that tank only gets like Saccharomyces cerevisiae?
Of course, yes. And also at the same time, there's a possibility that you don't succeed. Yeast is not particularly, I'm thinking about the word to use there, it's not particularly recalcitrant in the environment. So if I wanted to kill yeast and I'm thorough with what I'm doing, I can kill it off. If it doesn't mean that you can't have anything lingering around. But when I work with yeast in my culture, in my tank, I find that for the one I use in the lab, I used to use in my former lab, I find that it's rare that I finish a process and I sanitize and sterilize that I carry over what I had before. But my system in my former lab is smaller compared to a much bigger system. Now, the bigger, the more complex, the likelihood that something can linger. But again, the bigger also, the higher amount of heat you're going to put through the system to be able to kill off things. However, nothing is absolute with this process when it comes to sterilization. There remains that possibility. But I find that you're likely to find bacteria to linger in the environment more than yeast. Some of them sporulate more, and because of that, it's not as easy to kill them off. But yeast, you can get contamination. It depends on the skills of the people doing it. If they're not very thorough, don't understand it, or don't know how to do it, there's a possibility that you can linger, one of them can linger in the environment.
Okay, so Victor, can we talk a little bit about the growth aspects of some of these hybrids? Are you finding that some of these hybrids have similar growth aspects to normal brewing yeast? Because since fermentation is totally linked to yeast growth, does this present, in your opinion, a problem?
Well, each time we do hybridization, you find that you'll get multiple strains out of it, and some will grow poorly, some will grow well, some will grow extremely well. So it varies. And the ones that grow extremely well might not have the characteristics you want, and some will. So it's a matter of, it takes a lot of work to be able to screen and find exactly what you're looking for, and testing to ensure that it gives you what you want. And then testing over time to ensure that it's stable in the long term. So for sure, it can be done, but it involves an enormous amount of what I call physical labor. And it's beyond science. It's not, the hybridization part is the science, being able to screen and isolate and get what you want. I have a student working on a project now where we have about probably 500 different strains we're looking at, and it's going to take years. That student will do their part and go, another student will do their part. Over years, we'll find, we're already seeing some exciting new species or strains, we're interested in for different things we're doing, and some that are pure junk we're not even looking at again at this point. So whenever you do this, you get a wide variety of strains coming out of it. It takes time to sift through all of that to find the ideal ones you're looking for, and then test them over time to ensure that they are stable. For sure, you can find some that will grow well and hopefully have the brewing characteristics you want, flavor-wise and alcohol-wise. And in some cases, you find a whole lot of them that are just pure junk.
Thank you.
You're welcome.
So I have a potentially really nerdy question for you, but the discovery of the hybridization of Saccharomyces cerevisiae and Eubianus, I believe happened at University of Wisconsin with some other people working from Argentina. And Dr. Chris Hittinger presented on that. I think it was like 2011 or 2012 that he came out and gave a talk. And at the time, I might have the numbers flipped, but I thought he said that there was a 7% genetic mismatch between Cerevisiae and Pastorianus, and that Eubianus filled that gap. But then in your talk, you said that it was 99.5% Eubianus, and I'm wondering if that's just the result of further genetic investigation and genomics and really like dialing that in. Is that where that came from?
Yeah, absolutely. Over time, it was thought it was the other way. The recent study has shown that it's predominantly Eubianus than Cerevisiae. And that's why it thrives at lower temperature. While Cerevisiae likes higher temperature, Eubianus likes lower temperature. And the genetic study itself, I don't think it had been sequenced fully at the time and annotated at the time Chris gave that talk. I looked it up just a few days ago, and it's just about a small fraction that is Cerevisiae. There are a few genes actually that Pastorianus lifted from Saccharomyces cerevisiae.
Okay, that makes a lot of sense. So now you just have a much more complete map of how that looks. That's fascinating.
Yeah.
Has there been any work to reclassify different yeast strains, such as like Saison yeast, like to say this isn't really Cerevisiae, this is something else?
That's constantly happening all the time, and sometimes there are disagreements about it, and it takes a while to streamline things. Where one group thinks is this, another group thinks is this. But the beauty of it today is that genomics is allowing us to use the gene level to be able to classify and reclassify yeast. It used to be based on just how they look, and that is very, very deceptive at the end of the day. But genomics is allowing us to nail things down to the least level and say for sure we can tell that this is this or that. So there's a lot of classification and reclassification going on in the yeast world. And some of it is still controversial. Some of it we've been able to agree. Scientists have been able to agree as to which way that should go, but it's still happening a lot.
Okay.
I have a question on the Diastaticus yeasts. The majority of the ones that I've come across are all POF positive. And you're talking about hybridization, combining two, yeasts through hybridization. Is there a way through genomics to take a Diastaticus yeast that has, good fermentation temperatures and a flavor profile that is okay, except for that POF trait, and get rid of that POF trait to make it POF negative?
It's possible in so many ways. Some of them are acceptable in the brewing world, some of them are not. One way to do that is we can use CRISPR, go in there, remove the gene we want, edit it out, put the one we want. But once you do that, nobody wants to use that in brewing. That's one. Two, hybridization is another way you could try, but hybridization is you hope that what comes out of it will eliminate the traits you're not interested in. And sometimes it doesn't. You can have two distinct species existing or coexisting together and retaining their original properties, in which case you still have the off flavor you don't want, or you don't have the flavor that you want. So you have to do multiple hybridizations to be able to come up with something that could eliminate that. So it could happen, but it will take a long, long while to figure that out. One easy way to do it is to edit the gene responsible for that out. But the moment you do that, it's no longer eligible for brewing purposes.
Hey, Michael, just use Chico and add startup fermentation dry hops.
Sorry?
Some AMG.
You don't even need to. The
Yeah,
hops will supply the enzymes.
I was thinking, that's what, we could do that. I also have a yeast that if you ferment it at a lower temperature, that POF positive is kind of low. So,
And the higher temp mashing can offset that too. Evidently, all of the Guinness yeasts are POF positive, and you don't have phenolic flavors in their beers.
Oh, maybe that's all the dark. I use that for my Russian Imperial Stout sometimes. It makes a really tasty beer.
Yeah.
curious, there are other competitive factors other than, what carbohydrates a yeast can ferment or the initial inoculation rate. There's multiplication rate, but I'm curious if you're looking at killer factor of yeast strains. It's not something we talk about a lot in the beer industry, but it's more maybe a wine industry thing, that some yeasts are producing a protein that suppresses others. So curious if you're looking at that.
I'm not looking at that study-wise, but I do know that it still does exist. Yeah, it's more predominant in wine production. Some of them produce proteins, peptides that will affect other yeast in the environment. I think just like us, they have their home, someone else is taking over. They don't feel comfortable all the time about who's taking over. And sometimes it's down to what the new yeast is producing as well, that they are sensing that is not natural to them. And it elicits them to produce something to fight back. They just know there's an enemy in the environment. They produce bacteria to just attack them. Again, we've not seen a lot of that with these strains, but we've not paired them as widely as possible. The more of that you do, the likelihood that you could find something like that happening. I've worked with a few combinations in the lab, and they tend to attack each other. In fact, they tend to produce phenolics as compounds to attack whoever is growing in the environment. And the more phenolics you have, of course, the more off flavor you're going to get. So yes, sometimes I do have two species that don't like each other at all, that they make compounds that are just not good brew-wise.
Very interesting.
All right, I'm just curious. What are the two most antagonistic species of microbes that you found with each other? Yeah.
I've looked at a lot of them, both yeast and non-yeast fungi generally. I find that the more filamentous fungi are deadly. They don't like to have anything in their environment. I would say yeast are more tolerant, but once in a while, I find that the Diastaticus species tend to produce phenolics if you grow them with something else that is not native to them. They might produce a lot of phenolics, and those phenolics affect the cell membrane of both themselves and whoever is any other yeast grown in that environment. So yes, the Diastaticus, I would tell you, can be quite nasty sometimes.
Royal Rumble.
But Joe, are you thinking like a West Side Story kind of thing, but just with yeast?
Right. Or like a WWE event where like 30 wrestlers get in the cage.
And some of them make acids as well. Apart from phenolics, some of them just make more acids. They feel like if we can drop the pH further down, it's gonna hurt with whatever species is growing around here. Everyone is adapted. Every yeast is adapted to a particular sort of, I would say, antagonistic mechanism. Some use acids to be able to fight. Some use phenolics to be able to fight. Some don't have the capacity at all. So again, you can find one that makes right flavors, but the moment you bring someone else in the environment, they just begin to make too much of another kind of compound that's too much for the other guy to handle. And in some cases, both of them will die because of that. So yes, it does exist. And even with the protoplast fusion I talked about, that's why sometimes they exist for a while and they disintegrate, because you have that genetic crosstalk that is not comfortable for both of them, that they attack each other. But again, sometimes you find two that will blend in like nothing ever happened and just grow and continue to be together.
Awesome. All right, so it's about 7:20. I think that's thank you very much, Victor. We really appreciate your talk tonight.
Thank you.
Great talk.
Absolutely. Thank you very much.
Real pleasure. Thank you so much. Hopefully someday I'll come have a beer with you guys when we don't have to be on Zoom.
That's agreed.
All right.